Alright guys, so this video is going to cover our next lesson in genetics. So far we've done just kind of our basic Mendelian inheritance and some other patterns of inheritance, and we've looked at some Punnett squares. And so what we're going to do now is we're just going to kind of wrap up some of our inheritance information. And then in genetics we're going to have one more set of notes over fruit flies, and then we'll be getting ready for our test. So what this lesson's about is just about inheritance. We're also going to take a look at some genetic information that's specifically related to humans. And so one of the things that we've talked about over the course of genetics is that we have 46 chromosomes. And how do we know that we have 46 chromosomes? Well, we know that because we can do a karyotype. And so we actually looked at one of these in, I think, our genetics lesson. But a karyotype is just a picture of an organism's chromosomes. And so we could do this for any organism. But what you can do is you can take pictures during mitosis. Remember that the chromosomes are only visible during the stages of division. We see the chromosomes for the first time in prophase. They line up in the middle in metaphase. They're pulled apart in anaphase. And so a lot of times what they'll do is they will kind of pause mitosis with chemicals, and then they're able to kind of take a picture under the microscope of the chromosomes. So sometimes it'll be like in metaphase and there's still going to be X's, or sometimes it could be in anaphase where they've been pulled apart and they're just single chromatids. But essentially once they take a picture, they do a cut and paste kind of job. And so they create this picture called a karyotype. Now the karyotype can give us some information about the number of chromosomes, if the chromosomes are normal. And so what we can do is we can determine things like the sex of an individual, and we can also determine if there are any genetic disorders in terms of chromosomal issues. We can't see a single mutation like an A instead of a T, but we can look at missing chromosomes, missing pieces of chromosomes, extra chromosomes, things of that nature. And so these are both examples of some real human karyotypes. You can see that these were taken after those chromosomes were pulled apart because they're just single chromatids. They order them from largest to smallest, with the sex chromosomes last. You can tell that they go together because they're the same size and they have similar banding patterns. So if you remember, this is what we call our homologous chromosomes. And so as you kind of go through, you can see that they're arranged from biggest to smallest until you get to this last pair. And so in a karyotype, some important information that you will find are your autosomes, which are pairs 1 through 22. Now, this is something that you need to understand because when we talk about inheritance, we will describe traits as either autosomal or we could describe them as sex-linked. And so if it's autosomal, that means that the genes that determine that trait are somewhere on chromosome pairs 1 through 22. And what's significant about autosomes is that everyone, doesn't matter whether you're male or female, everyone should get 2 copies of the same chromosome. 2 number 1s, 2 number 2s, they're the same size, they have the same genetic information. So it doesn't matter whether you're male or female, everyone's getting a copy from mom and a copy from dad of the same chromosome. Now, pair 23 is a little bit different. Pair 23 is what we refer to as our sex chromosomes. And these will be different depending on whether you are male or female. If you are a girl, you got an X from mom and an X from dad. And if you're a boy, you got an X from mom and you got a Y from dad. And so when we look at traits that are on the sex chromosomes, they are not necessarily going to be inherited in the same way between male and female. So autosomes, everybody has the same chances of getting a particular trait or not getting a particular trait. It all depends on what their parents give them. We don't have to look at them in terms of male or female. Everything we've done so far has been autosomal, and we've seen how we write our genotypes. We don't have to take into account whether they're XX or XY. We can just simply use capital A, lowercase a, big B, things of that nature. And so another thing that we can do to study genetics is we can use pedigrees. And pedigrees are essentially kind of like a genetic family tree. They can be used to trace a trait through a family. Now, I'm not sure why, but this word often gives students some problems because when you see questions about pedigrees, it'll ask about the trait being traced. And so a pedigree can trace any trait. It can be a dominant trait, it can be a recessive trait, it can be a disease, it can be something normal, it can be autosomal, it can be sex-linked. So you could go home and you could make a pedigree for curly hair in your family. Or you could go home and you could do a pedigree for freckles. Or you could go home and you could do a pedigree for some type of disease. You could do something like hemophilia, a bleeding disorder. Or you could do something that might run in your family. If it's something that is maybe certain types of cancer or something like that. If it is a trait that is inherited, then you can literally trace it through any family. And so what it does is it just shows the people in your family and whether or not they have certain characteristics. And so you can analyze a pedigree to determine maybe how a trait is inherited. You can also possibly determine the genotypes. If you know how it's inherited, then you can usually figure out the genotypes of most family members. And so what a pedigree does is it uses symbols to represent the people in the family. So circles are females, and remember that females have curves, okay? So anytime you see a circle, that's a female. And then males are squares, okay? So if you have a circle or a square that is unfilled, that means that they do not have whatever trait it is that you are tracing. If they are filled in, that means that they do have the trait that you are tracing. Now, in some pedigrees, it depends on the inheritance pattern, but in some pedigrees, you will find carriers possibly. And a carrier is a heterozygote. Okay, so you may want to write this out to the side. A carrier is a heterozygote. And something that's really, really important to understand is that you only see carriers if the trait is recessive. Okay, there's no such thing as a carrier if you have a dominant trait. Okay, so let me kind of explain this for a second. If you have a heterozygous person, let's say they are capital A, lowercase a, okay. Now, if we were looking at a dominant trait, all right, this person here, capital A, lowercase a, would have the dominant trait. So if we were tracing a dominant trait and this was a person's genotype, then they would be completely filled in, okay? Now, if the trait was recessive and this was a person's genotype, they do not have the recessive trait, okay? So we would not fill them in because they do not have the trait. However, they do have the recessive allele. And so sometimes, maybe not always, but if it's a recessive trait, you may see carriers. And what that means is they carry the recessive allele, but they do not physically have the trait. So notice they're only half filled in. So they carry the allele, they could pass it on to their children, but they do not physically have that trait. So once again, you only see carriers if the trait is recessive. Okay, so that's kind of a clue to the inheritance pattern. Now we also represent relationships with lines, and so I'm going to go to the example pedigree to kind of explain that. So here's a family. Now up here they're telling us that it's X-linked recessive, and we're going to get to what that is, and this pedigree will make a lot more sense once we get to recessive X-linked traits. But for now, we just need to understand how to read the pedigree. So this would be generation 1. Sometimes you'll see Roman numerals like this. Generation 1, generation 2, generation 3, and generation 4. Sometimes you'll see numbers under the individuals, like a 1, 2, 1, 2, 3, 4. And so that allows you to identify individuals within the pedigree. Pedigrees can vary. But what you need to understand here are the relationships. So here we have a man. Okay, he does have the trait. In this case, we know that that is red-green color blindness. Now, a horizontal line represents a marriage or a mating. And so this is maybe his wife or someone that he mated with. Notice that she does not have that trait. Okay, and so the vertical line branches down to their children. And so you can see they have a son here. He does not have red-green color blindness. They have a daughter who is a carrier. They have another son who doesn't have the trait. They have a daughter who is a carrier. And then they have another son that does not have the trait, okay? So this person here would be their son-in-law, and this person here would be their daughter-in-law. Then you can see they went on to have children. So this couple had 3 children, and this couple had 3 children, and their daughter got married, and their daughter got married. And so that's kind of how you read a pedigree. Now, once again, this one kind of is telling us what it is, but let's say we didn't have this title up here, and you just saw this pedigree. There are ways to look at this and determine how a trait is inherited. So like the fact that there's carriers, that would automatically tell us that the trait is recessive. We could also determine that it was X-linked based on the inheritance pattern, and we'll come back to that here in a little bit. So you do have in your problem set, we don't really have any to practice, but in your problem set you do have some pedigree questions, and so you need to be familiar with those. You should be able to identify their genotypes. You should be able to kind of determine the inheritance pattern, and you should be able to answer questions like how many people in the family have the trait, or how many people do not have the trait, or how many males have the trait. So maybe we'll take a look at those whenever we look at blood and sex-linked Punnett squares. All right, so moving on, just some general vocabulary. Once again, we're kind of looking at human genes, and so a genome, this is just a general vocab word that you need to know, but genome is your complete set of genetic information. It's all of the genes for a particular species. So we have something called the human genome. Now, we don't, each of us as individuals have every possible gene because it's dependent on how they're inherited. But as a human species, there's a genome of all of the genes and all of the chromosomes that you will find. And so one of the things that's been ongoing over many years is trying to figure out what all of our genes are, what they all do, where they all are. Now, I know this was a long time for you guys, but this year is kind of significant to me because this is the year that I graduated high school. And so it was kind of a big deal back then. And so the Human Genome Project was completed, and essentially what they were trying to do is identify pretty much our genetic code. It was pages and pages and pages and pages of letters, the A, T, C, G, over and over and over again. We have billions of letters in our genetic code. And since then, we are still trying to decipher it. Many of our genes have been identified. We have areas in our genome that we know are not genes, they are maybe regulatory sequences, and then we also still have some unknowns. So this is something that is still in the works today. So we now have our genetic code kind of written out. Now we're just trying to figure out what all of it does. Okay, so that's, like I said, is still a work in progress. Now, some of the first genes that were identified were blood typing genes, and you're going to have some blood typing problems to do tomorrow. And so there's really 2 main components to your blood type. There's actually more, which is why if you get into organ donation and things like that, there's a lot of markers that have to be the same. But in terms of your basic blood type, we usually refer to it in terms of 2 parts. We have the ABO blood group, and so that's the component where you're A, B, or O. And then you have the RH blood group, and that's where you get the positive or negative. Okay, so it's actually 2 separate genes that determine those. They're both inherited independently, and so the ABO blood group is just the letter portion, and the RH blood group is the positive negative. And so we're going to take a look at each of these. So the RH blood group, first off, it's named for the rhesus monkey because that's where it was discovered. And so the RH blood group is whether you are positive or negative. Now what that really means is, do you have a protein or do you not have a protein? All right, so I am O positive, and so that means on my blood cells, I have a particular protein that a negative person does not. And this does play into blood donation and receiving blood. Because hopefully you know that your body does not like things that do not belong. So if you're a positive person, you're good and you can receive pretty much any type of blood. But if you're negative, you can't receive positive blood because your body's not going to recognize that protein on those blood cells. Now this is a very simple inheritance pattern. There's a single gene that determines it. There are 2 alleles. Positive is dominant and negative is recessive. Okay, and so essentially this would be like a capital A, this would be a lowercase a, and just like all of the regular old traits that we've done so far, you could be homozygous dominant and have positive blood. You could be heterozygous and have positive blood, or you could be homozygous recessive and your blood would be negative. Okay, so like I mentioned, it just simply means that you have an extra protein on your blood cells if you are positive. Now the other component is the ABO blood group, and so once again we have a single gene, but this time there's actually more than just two possible alleles. You have an A allele, you have a B allele, and you have an O allele. Now it doesn't matter to me at all how you use these, but I do want you to know that the official way to write out these alleles is with a capital I and a superscript if they are dominant, and a lowercase I for the O allele because it is recessive. I just simply use A, B, and O because I know the relationship, okay? So this is a codominant relationship. A and B are both dominant, which is why if you get both, you see both, and that's how you get the blood type AB. Now O is recessive, and so the only way to be O blood is if you have two O alleles, okay? This is another example that just because something is recessive does not mean that it is less common, because type O blood is the most common, okay? O is the most common. Technically, O positive is the most common. And then as you go down, it's O, A, B, and AB, with negative being less common than positive blood. Now, the way this works is, once again, when your genes often determine proteins, and so these alleles determine which proteins or antigens are on your blood cells. So like if you're type A, you have the A antigen. If you're type B, you have the B antigen. And if you're type O, you have no antigens, okay? Now, our body, when we don't recognize something, we will produce antibodies against it. So antigens will trigger the production of antibodies if they are not recognized. So this is why, like if you're type A, you cannot receive blood that has a B antigen because your body would not recognize that protein. So A people would produce antibodies to B, and B people would produce antibodies to A. It's kind of backwards like that. So this is why we have to be careful when we are getting blood or giving someone blood because we don't want to have these immune reactions. So just to kind of summarize, okay, you have, if you're type A, you have the A antigen, you produce antibodies to B, and the genotypes that can produce type A blood are here. If you're type B, you have the B antigen, but you would produce antibodies to A. These are the genotypes that could give you type B blood. If you're AB, remember those are both dominant, so your genotype would be AB. You have both antigens, and so you would not produce any antibodies. And then if you are type O, you have neither antigen, but you would produce antibodies to both. And the only way to get O is to have two O alleles. The only way to get AB is to have one A and one B. And we'll look at that further when we do blood Punnett squares. Now what can happen is if you were to give someone the wrong type of blood, and this goes for both ABO blood group and positive negative, it could lead to something called agglutination. This is actually how we determine our blood type, is we add antibodies to our blood to see what happens, to see if they clump up. And so O doesn't have either antigen, and so this is why we can give O to anyone. Now technically it's O negative. O negative is the true universal donor because anybody can receive that and they will not produce antibodies because there's no antigens. Now AB, because they don't produce any antibodies, they're what we call the universal receiver. And technically it is AB positive that is the true universal receiver. All right, so another type of inheritance that we're going to look at is sex-linked traits, or sex-linked genes. And so we mentioned earlier that on our karyotypes, our first 22 pairs are what we call autosomes, and everybody gets 2 of the same chromosome. Everybody has an equal opportunity of having a particular disorder or not having a disorder. It all depends on what your parents give you. Now, with sex-linked genes, these are going to be genes that are on the sex chromosomes. And really, it's normally on the Y. Okay, there's X-linked traits and there's Y-linked traits. Most of them are going to be X-linked. Okay, and so the Y is little bitty tiny. Okay, pretty much what the Y does is it makes a male a male. There can't be essential genes for being a human on the Y chromosome because females don't get one. So while there is genetic information on the Y, it is small, and there's just a few genes that mostly make a male, okay? So normally when we're talking about sex-linked traits, we're typically talking about X-linked traits, which means the gene is on that X chromosome. Now I want to go back just a second. I'll try to make it quick, but I just want to show you. So this would be a male right here. You can see that this X chromosome is much larger than that Y chromosome. So when we're talking about sex-linked traits, the gene is physically on that X, okay, if we're talking about X-linked. So you see how here the male only gets one copy of that X. The female will get two copies of that X, okay? And so when we're talking about sex-linked traits, there will be a difference in inheritance between male and females. So a lot of the traits that we will do problems on are sex-linked recessive. And there's some common ones here like color blindness or hemophilia. This is technically red-green color blindness, like we saw in the pedigree earlier. Hemophilia, Duchenne muscular dystrophy, and all of these are going to be more common in males. And the reason why is because it only takes one recessive allele for a male to have an X-linked trait. A female gets two copies, so a female has an opportunity to cover it up. And we'll kind of look at that inheritance further when we do the Punnett squares. Now, just because a female has two X's does not mean they get a double dose of every gene. Clearly, a man can get by with only one copy of that X chromosome, and so it's not necessary to have a double dose. And so what will happen is that in female mammals, in our cells, randomly, one of our X's becomes inactivated. That way we're not receiving a double dose of those genes. And so what will happen is that one of our X's will kind of super condense up into what's called a bar body. And so under the microscope, this is what it looks like. And essentially it's just an inactivated chromosome. So in each one of our cells, we're only going to be expressing one of our X chromosomes. Now, even if we have some of those recessive disease alleles, we're still fine because probably about 50-50, half of our cells are going to have normal and half of our cells won't. So we're still going to make the proteins that we need. So really females won't necessarily suffer the effects of a disease, even if some of those X's are inactivated, or even if disease X's are activated. Now, sometimes this could lead to what we call a mosaic effect. I don't really know of a lot of specific examples in humans, but this is what leads to the coloration in calico cats, is that because different genes are activated in different cells, you can see some different phenotypes that might be expressed together, and that's what leads to that coloration of those calicos. I did have a student tell me one time that they had a male calico, but typically if you see a calico cat, you know it's a female. All right, we're also going to talk a little bit about genetic recombination, and we've really already talked about this, but recombination always equals new, okay, and different. Recombination is when you get offspring that are different from the parents, and this is essentially what sexual reproduction does for us. This goes back to meiosis because of independent assortment and crossing over, which we talked about very heavily back in meiosis. All of the offspring that are produced by sexual reproduction are going to be products of genetic recombination, and the offspring will be different from their parents. And so when it comes to our inheritance and recombination, we mentioned under Mendel's laws, the law of independent assortment, that each trait is inherited independently of each other. That's not necessarily true. Like I told you before, Mendel got lucky with his pea plants, but technically it's the chromosomes that are independently inherited, not each individual gene, okay? Now, we do know that each gene has a specific location, a loci, or locus. Locus is one and loci is plural, I believe. But each gene has a specific location on a chromosome. It's going to be in a certain position on a certain arm of the chromosome. And so it is the chromosomes themselves that actually undergo independent assortment. not each of those individual alleles. So there are some other ways that we could not get those Mendelian ratios, and it could be if the genes are linked. And so linked genes are genes that are on the same chromosome. So a lot of times they will be inherited together. The only way that they would not be inherited together is if crossing over caused them to be separated, okay? And so it's using these ideas of linked genes and crossing over and expected ratios that we can kind of create chromosome maps. Because what can happen is if we expect them to be inherited together and they are not, if we get a different result in the reality, then we know that somehow those genes must have been separated, and it is crossing over that would have done that. Okay, so an example of linked genes in humans is red hair and freckles. Now, it doesn't mean every redhead has freckles, but those two traits are often inherited together because they're on the same chromosome. So using this concept of crossing over and not getting expected ratios, it's one way that has allowed us to create these gene maps. Now that we can sequence DNA, we can look at genes and we can identify their sequences and where they're located. But when we first kind of started studying the location of genes on chromosomes, it was kind of done with this concept in the background. And our next lesson is going to be on fruit flies, and a lot of what we know, like these genetic maps, comes from fruit flies because they have a very small number of chromosomes and a small number of genes. Now, what you need to understand is just the concept of how we can look at location of genes based on crossover rates, okay? So keep in mind that if you have linked genes, that means they're on the same chromosome. So oftentimes they are inherited together. So you would have a specific expectation of a genetic outcome. This is what Punnett squares do for us. We can predict what we would expect. Now, we know that that doesn't always happen. And so when it comes to linked genes, the reason we may not get what was expected is because crossing over could have separated those linked genes. And so what we can do is we can look at a percentage of crossover. Now, this is going to be hard to kind of show on the lesson, but let's say this is my chromosome, and then I have my homologous chromosome here. All right, I'm going to get a pen. So let's say this is gene number 1 here, and this is gene number 2. All right, and then we're going to have the same thing over here. Now, you can see that there's some decent space between these genes, okay? So there's a lot of room for this crossing over to potentially separate them. Now, what if I had another gene that was right here below this one? Okay. Now, in this instance, there's a very good chance that if crossing over occurred, these two genes would stay together, okay? So the whole concept behind these linkage maps is that the closer two genes are, the less likely they'll be separated by crossover. The farther apart they are, the more likely they'll be separated when crossing over occurs. And so what they can do, and they really don't do this with humans because this would involve test crosses, but with fruit flies, what they can do is they can kind of look at the crossover rate. And they came up with their own little unit of measurement, because remember, chromosomes are tiny, so map units. So whatever the percentage of crossing over is, or recombination, that means those genes must be further apart. Okay, so let me kind of show you the picture here. So here's a gene for body color, and here's a gene for eye color. Now, according to our map, these are 8 map units apart, okay, because they have an 8% crossover rate. The eye color and wing size gene, they have a 9% crossover rate, which means that they are 9 map units apart. Okay, so whatever the crossover rate is, that is our map unit distance between those chromosomes. Now you can see that body color and wing size have the longest distance So they have a larger crossover rate. So the more that they cross over, the closer they must be, okay? And that's essentially how we determine the distance. So the closer they are, the more likely they will stay together, okay? And the less likely crossing over will separate them. So the bigger the crossover rate, the further the distance, okay? I hope that makes sense. I know that's kind of hard to explain through the video. I did want to show you up here that this is an example. We could be doing more Punnett squares, but this is an example of a Punnett square using linked genes. So we would expect certain genes to be inherited together, and if they're not, that would tell us that recombination occurred. All right, a couple other things before we wrap this up. Not all of our traits are determined by our nuclear DNA. So when we talk about our 46 chromosomes, that is going to be the genetic material that is in our nuclei of pretty much every cell in our body. However, there are some traits. We do have another type of DNA that you can find in your cells, and that is mitochondrial DNA. Now, we don't have chloroplasts, but we know that mitochondria and chloroplasts both have their own genetic information. They both have their own DNA. And so there are some traits that could come from mitochondrial DNA. Now, for the most part, your mitochondrial DNA comes from your mom. And if you ever watch like forensic type shows, a lot of times this will be mentioned. Mitochondrial DNA can like last longer than nuclear DNA. So a lot of times it's used maybe like in bones or something like that. But the mitochondrial DNA can help them trace family lines on the maternal side typically. Remember how you get one really big egg and the sperm pretty much just drops off the chromosomes? So the mitochondria that you have were essentially coming from the mitochondria that were in that initial egg cell. So mitochondrial DNA can show us traits that are inherited from the mother. And there are diseases and things that are linked to mitochondrial DNA. All right, and the last thing we're going to talk about are mutations. Now we're going to focus on these gene mutations more specifically when we get to protein synthesis, but there are different types of mutations. Now, a mutation in general is just any change to DNA. Now, that mutation can be at the gene level, and most of these are just going to be basic point mutations. This is where you have a difference in letters, where maybe I have an A, you have a T. These are just going to be either single letter changes or a very small change in one gene, okay? Now what that does is that can affect the protein that's produced. And like I said, we're going to look at these in more detail when we get to protein synthesis. But these are what we call SNPs, single nucleotide polymorphisms. And most of our uniqueness and differences as humans is going to be in these just slight variations in our nitrogen bases. Where I have a C, you might have a G. And so a point mutation can ultimately change the protein. It might keep it the same. It could change it for good. It could change it for bad. It could be lethal. It all depends on where and what gene. But a point mutation essentially affects one gene and one particular trait. Now, another type of mutation could be a chromosomal mutation. And this is where you have it at the whole chromosome level, where you could have pieces of chromosomes or whole chromosomes that are affected. And in this case, you're going to have many genes and therefore many effects, okay? It's not necessarily one protein that's being affected, but several proteins. And so a lot of times the effects of a chromosomal mutation, there will be more traits that could be affected by those. So you could have like a deletion of a whole or a part of a chromosome. You could have certain pieces of chromosomes that are duplicated, like for instance, Huntington's disease is due to a duplication of a particular gene, like 30 times more than normal. You could have inversions where the gene gets flipped, and so now the genetic message is backwards. You could have a translocation. This kind of looks like crossing over, but it's where a piece of one chromosome breaks off and attaches to another. And so now you have extra genetic info in one sequence and missing genetic info in another. And so all of these could lead to all different sorts of genetic disorders. It really just depends, once again, on where and what, which chromosome was it, and which genes. So here's an example of what those might look like. Okay, so a deletion, a piece is removed. Duplication, you have extra copies of genes. inversion, it gets flipped. You could add a piece to another chromosome, or you could have a translocation where it breaks off and attaches to another. Now, these chromosome disorders could lead to an abnormal chromosome number, and this is caused by something called non-disjunction. So in meiosis, and this can happen either in meiosis 1 or meiosis 2, but if chromosomes don't separate properly, you could end up with some gametes that have extra chromosomes and some gametes that have missing chromosomes. And so aneuploidy is just when you have an abnormal chromosome number. Typically, the ones that you hear about are either trisomies, which is where you have one of a certain chromosome instead of two, or, I'm sorry, I said this backwards. A trisomy is where you have one extra chromosome, so you have 3 instead of 2. A monosomy is where you only have 1 chromosome instead of 2. You typically don't hear of a lot more than that because it would probably be detrimental and the baby would not survive. But aneuploidy could be something like 47 chromosomes or 45 chromosomes. And then you could have polyploidy where you have extra sets. Now, this is not common in animals at all. We don't see this in humans. It's normally fatal in animals, but it's pretty common in plants, and a lot of times it makes them bigger and stronger. But this would be having like a whole extra 23 chromosomes or a whole extra 46 chromosomes. And in animals, that just leads to too many genetic differences, and so it never results in a living offspring. So this is what non-disjunction looks like. The chromosomes just don't separate properly, so you end up with extra chromosomes in some gametes, missing chromosomes in the other. And of course, if any of these were used to create an offspring, they're going to have an abnormal chromosome number. Now, these are just a few common examples of aneuploidy. You have Down syndrome, which is also called trisomy 21. You have Turner syndrome, which is a female that only has one X, and then Klinefelter syndrome, where you have a male that has an extra X. A lot of times these people don't even realize they have it. It's relatively mild. But there's other examples. Now, you don't see examples of all chromosomes having extra or missing because, once again, sometimes these genetic abnormalities could be fatal and they could lead to incomplete development. And so some of these disorders they never even come about because we don't have a living person to identify that abnormality in. So a lot of times you'll just see a few common examples. There's a couple others like Edwards and Pateau syndrome. But the main thing you need to know for now are the different types of mutations, like point mutations versus chromosomal mutations. And that is going to wrap up our human heredity. Tomorrow we're going to take a look at the blood and sex-linked Punnett squares and maybe a little bit about pedigrees. You will be having a quiz here in a few days, and it's going to cover the human heredity notes as well as all of the Punnett squares that we've covered since the monohybrids. So you'll have dihybrid, you'll have other genetics problems, blood and sex-linked, and some pedigrees. I hope you guys have a good day. If you have any questions, just let me or Miss Petty know. And be sure if you need any help on genetics problems, be sure you get the help that you need. All right, y'all have a good one.